Hello everyone, welcome to the webinar on the phylogenetics module of Biopython. The webinar is organized by Scientia Bio. Scientia Bio is a one-stop knowledge service provider for pharma, CROs, BPO and academia. It was established in the year 2010 with its headquarters in Bangalore. Our website address is www.scientiabio.com. We offer a lot of educational courses in Basic Perl, BioPerl, Python, BioPython. Apart from that, we also offer online live training on topics like computer-aided drug design, protein structure, biostatistics, clinical research, basics, and advanced, etc. We have been fortunate enough to work with several industry giants like Novartis, Quad One, Quintilis, Accenture, and lot many such client lists adorned our. Today's agenda would be we would be discussing on what is phylogenetics, why phylogenetics, a module called as Biopython, a module called as Biophylo from the Biopython programming language. We will have generic case studies. We will also have a look at Philo XML, and in the end the session would be open for question and answers. Before we begin the session, I would like to introduce myself. My name is Pallavi, and I have around say six years of experience in the field of bioinformatics. For some time, I've also worked in US in the University of Virginia Tech, and I have around 4 years of experience in conducting trainings and project consultation services on BioPython, BioPerl, BioJava, etc. Before we begin the session, may I request you to please introduce yourself. You can type your introduction on the question window panel which you can see on the screen. Also, I hope that you are all able to see the screen properly and my voice is clear to all of you. So please kindly introduce yourself. Kindly note that during the webinar, since you have joined as an attendee, you will be put on a mute mode, and during the question and answer session, I will unmute you if required. So you can raise your hand and tell me that you want to be unmuted and I can unmute you. Meanwhile, I request you to please type your introduction in the questions window. I would like to know from where have you joined this webinar and a brief about your experience in this field of bioinformatics. Hi, Leela, can you please introduce yourself? Hi, Sebastian, can you please introduce yourself? I know you don't have a mic. You can type Sebastian. Just type a brief about you. As in, from where have you joined the webinar and how many years of experience do you have in the field of bioinformatics? And what's your area of specialization? Are you working on something currently? Okay. Glad to know about that, Sebastian. Okay. Amazing. So you already have an experience with phylogenetics. That's great. Leela and rest others, can you please introduce yourself? Okay, moving on. Evolution is effectively a process whereby populations are altered over time and may split into separate branches, hybridize together, or terminate by extinction. The evolutionary branching process may be depicted as a phylogenetic tree, and the place of various organisms on the tree is based on a hypothesis about the sequence in which evolutionary branching events occurred. So, phylogenetics is effectively the study of the evolutionary history, development, and relationships among different groups of organisms. These are discovered through molecular data matrices based on nucleic acid sequences and protein structure. The result or the output of a phylogenetic study is a phylogeny, a hypothesis about the evolutionary history of taxonomic groups. A very famous diagram called Tree of Life is represented in here, which basically says that all the living organisms existing can be divided into three major clades: archaea, bacteria, and eukaryota. And phylogeny is a lot like reading a family tree. A clade is effectively a grouping that includes a common ancestor and all its descendants. So here in this tree of life, we can see that the different sequence genomes are generated in different color codes. The advantages of phylogeny are many. Phylogeny has helped in finding cheaper drug alternatives. So there are some articles available at the Berkeley site. Let's have a look at them. So phylogeny basically helped us figure out a very effective drug compound called as Taxol, which is very much effective in treatment of cancer. But earlier it was discovered to be very expensive from the organism derived. Phylogeny basically helped us to find an alternative, organ, cheaper organism through which this compound could have been extracted and is made possible to be available to millions out there. Phylogeny also helps us establish the evolution. So people studied about the spider web formation, cobwebs and more orderly webs, and they discovered in which order which behavior or spider web formation was discovered. Phylogeny is also an increasingly important tool in modern biology. It has been applied to solve problems as diverse as uncovering the origins of HIV, making conservation decisions, to testing hypotheses about long extinct organisms. A phylogenetic tree or an evolutionary tree is a branching diagram showing the inferred evolutionary relationships among various biological species or other entities. Their phylogeny based upon similarities and differences in their physical and genetic characteristics. Different types of phylogenetic trees are seen: rooted tree, unrooted tree, a bifurcating tree, etc. Some very specific algorithms are used for the construction of phylogenetic trees. They are generally classified as neighbor joining algorithm, maximum parsimony, and Bayesian framework, which mainly talks about hidden Markov chain models, which helps us figure out the phylogenetic tree. Many softwares are also available out there which help you construct these trees. So there are a lot of softwares, both web-based and something that you can install on your system, which help you fetch those trees. With the introduction of bioinformatics and availability of a large number of programming languages to support and provide different libraries to solve biological problems, for example, Perl offering BioPerl, Python offering BioPython, a large number of freely available tools to solve biological computations are made available. In one of them, and a very famous language known to solve biological problems is written in Python by an international team of developers called as Biopython. The Biopython license is very liberal and it's compatible with almost every license in the world. And a lot of information about it, rather a complete set of information about it, is made available in the biopython.org. Now, even if there are a variety of softwares already existing out there, why it is a need to learn a language called as BioPython, which has got a very huge support to solve biological problems, very competitive modules to handle large scale data sets, easy to use functions to process information from a large data, is because few of the advantages which we get is data security. We do not have to post our data on a public website which is secure, and we can easily keep such data within our laboratory bounds by implementing our own modules in a programming language to process our data. Customization of these modules as per our requirements. The softwares out there which are already created capture data, gather data, analyze data, and produce results in a predefined format. At times, you might be looking for something more. So, customization of resources is in your hands. You can customize the modules, the implementation of these modules according to your requirements. The results are most faster and accurate. You don't have to wait for 2 days, 3 days, or a week's time. Neither for that number of time your data would be hosted on a public domain site. Since change and the control is in your hand, the reliability and the robustness increased many folds. BioPython has a very interesting module called as bio.philo, which was created following the lead of sequence input output and align input output, and its main aim was to provide a common way to work with the phylogenetic trees independently of the data source format. The module bio.philo mainly provides four input-output functions: parsing, reading, writing, and conversion. Each function accepts a file name or a file handle, and the data can also be loaded from the compressed files. Different formats in which phylogenetic trees are created is supported by BioPhilo modules, which are Newick, PhyloXML, CDAO, Nexus, etc. So, creating our base of what we have understood by phylogenetics and the language BioPython module available bio.philo, we will now see some practical implementations of it. The very first thing which we are going to see is how we can generate a tree from a set of sequences in a FASTA file. So the process of generation of a tree from a set of sequences in the FASTA format are: we first should have a FASTA file available from any of the databases like PFAM, UniProt. So your sequence or your gene of interest or your protein of interest, you have that sequence available, you run a FASTA on it, blast on it to gather or find similar sequences, and you get hold of this FASTA file. Once this FASTA file is made available to you, you then have to do a MSA technique over it using a tool called as ClustalW, which basically helps you generate a dot alignment file, a multiple sequence alignment file, along with a Newick file. That's your tree in the .dnd format. The generated file, the .dnd file which gets generated, becomes an input to the BioPhilo module, which helps you draw the tree, perform different operations on this tree to process, analyze, information from this data source. So we should have our FASTA file available. Then we are going to run a cluster W on it to generate our alignment and DND files. And finally, we are going to use the BioPhylo module to make sense out of this data. So to solve this particular or to showcase you this demo, we are going to pick up a protein called as PF05371, and it is available in the PFAM database of protein families. It's basically a phage major code protein called as GP8 or B. And let's go and visit this link. So it is going to open your PFAM database for this particular protein entry. In here, we have availability of different sequences which have been found similar to this protein structure in the form of seed sequences, a smaller subset, full sequences in form of a larger subset of sequences which can be downloaded. Then going on to species, you have a sunburst diagram showcasing from where these sequences have been found and a taxonomic lineage interpretation of the different species involved. Now what we are going to do in here is we are going to have availability of the seed, a small subset of FASTA sequence in the form of seed.fasta made available to us. Using the BioPhylo module, we are going to generate a seed tree which should ideally match the tree shown case here in this particular website. So let's see how to do that. So our steps are availability of a .fasta file, availability of cluster w2.exe on the system so that we can do a MSA over the file, the output a .dnd file will be executed with the help of a BioPhylo module. So these are the steps which we will be taking into consideration. So we have got some examples available with us. So this is the code c.fasta file which we have downloaded from this website. So these are a set of around 7 sequences which are found to be similar to our phage protein. Using this file, we are going to generate a .dnd. So let's look at our program. From the BioAlign applications, we have to import cluster command line. So the cluster W is basically a software which gets installed onto your machine, and it can be run using a command line. Before writing down a program on cluster W, we should first check whether the cluster W installation is proper by executing it over on the command line of our system. Once you have done that, you can go ahead and use this cluster W into your program. So we first create a variable cluster W dot underscore exe where we basically say and provide the path where our cluster w2.exe is located. Okay, that's the first thing which we have to do. We have to make sure that the path where the cluster w2.exe is located is stored in a variable. Then we provide an input file path and an output file path. The input file path in here is available in the D drive location, and the name of the file is codec.faster. Then the output file where we want the output to be generated and saved in the .aln extension. Then we execute the command cluster command line, name of the exe, the path where the exe is available, in file equal to in underscore file, out file is equal to out underscore file. So at the runtime on the command prompt, this particular exe will get executed with these two input arguments, the in file and the out file. Let's run this particular program. So our program got executed pretty fast because our data set was small, and we got availability of our ALN file and a .dnd file. A .dnd file is basically our NuVic file. So let's have a look at it. So this is the phylogenetic tree in the DND format which gets generated. Now to make it much more meaningful to us, we are going to draw it using the bio.phyllo module. For that, we are going to open a function called as generate tree, and in this we are going to import the phyllo module from the bio module available in Biopython. We give a path to our DND file codec.dnd. We use the read command to read this particular file in the format NuVic, and we use the command phyllo.draw_sky, and we pass the tree variable to it, the tree object to it, sorry. Once you run the module, you will get generated a phylogenetic tree. If you look closely, this tree generated is exactly a replica of this tree, okay, made available here. So the same, using availability of FASTA sequence, a cluster W tool, and the BioPhylo module, we were able to generate a phylogenetic tree from a set of FASTA sequences made available to us. You can use the same process on any data set, a small, a big, a large, for any data point, whether a gene or a protein sequence, whatever is your interest area. Do you guys have any questions so far about what all we have discussed, or any observation, anything which you would like to share with me? Any comments, any suggestions? Please feel free to type any question in the question window. I'll try my best to answer it. Does Philo module have a facility to make a circular type of tree? Well, there are different tree drawing modules available like MAT, OLIB, PyOLab, GraphViz tools, etc. available, which does help you to generate a tree in a circular format. We can do that. Sure, you're welcome. Okay, moving on to the next topic. So we have seen availability of a phylogenetic tree in a format called as Newick, saved with an extension of DND. Now, it has been found that sometimes it is not possible to represent a lot of information about a organism or a sequence inside a phylogenetic tree using the Newick format, for which there is a need felt to generate and make available a language, a data representation technique which could carry a large number of information points regarding a sequence and can be represented in a manner which can be consumed by a large number of applications. So, a language called as PhyloXML was designed to describe the phylogenetic trees or networks and the data associated with it. It basically provides elements for the commonly used features such as taxonomic information, identifiers, branch length, gene duplication, events, etc. The XSD of the PhyloXML, which basically showcase the structure which a PhyloXML file can have in detail, is available here. So in its complete form, a PhyloXML file will start with the PhyloXML root node, will go on to the phylogeny node. The phylogeny node can have a name, ID, description, date, clade information. The clade can have its own sequence, property, relation information. And then it will have a clade node. The clade node can have its own name, branch length, confidence, width, taxonomy information. The taxonomy itself is a node which can be expanded, followed by sequence information. So a lot of information regarding a particular sequence or an organism can be captured in this PhyloXML and transferred and consumed by different applications. So we will see how we can generate a PhyloXML from a DND file. So remember that the Python also provides us a methodology to convert an exist format in a different format. So we'll see that. So we again import out the phyllo module from the bio module in Biopython. Then we make available from the phyllo XML inside the phyllo module, the phylogeny module. We pick the DND file and we give the XML file name as the same name.xml. In here, I am using the same name again and again, but changing the extension is because I want to keep it in a similar format. It is not necessary that the name of this file should always be codec.xml. It could be anything. Okay. Then once we have made the path available of DND in XML, I create my tree object by reading this file, DND file. Then I use the command phyllo.convert, where I convert this file made available in Newick format to an XML file in the PhyloXML format. And we simply then print this tree. So the DND formatted file is now converted into a PhyloXML as generated here. So if we go in backend and double click on the XML file which gets generated, we can have a look at the phyllo.xml contains the phylogeny node, then it contains clade. A clade node can contain another clade node. Finally, it comes across a clade node which has a name and a branch length associated with it. Again, sibling clade node with a branch length associated, and so on and so forth. So this information is represented to us now in an XML format. Now, we will talk briefly about Python BioPhylo objects called as tree and a clade object. The tree object is basically produced by the parse or the read function, and it is attached to the root attribute. A clade is basically a branch which has got clade specific information and a list of its own descendants. Both the tree and the clade are inherited from a BioPhylo module called as tree mixin class, which basically contains the implementation for methods that would commonly be used to search, inspect, or modify a tree or any of its clades. The code and the methods which are available for a tree mixin module is available in the Biopython org documentation. Here you can find out that a tree or a clade object can support some of the inbuilt methods like find any, find elements, find clade, get path, get terminals, common ancestor, count terminals, is the tree bifurcating. So basically such methods which are inbuilt to the tree mixin module and is true for the clade and a tree object in a Biopython module helps us to process and analyze information. So some of the generic methods which we can use are broadly into search and traversal methods. Search and traversal methods like get terminals, get path, information methods. For example, find a common ancestor, distance calculation, modification methods like collapsing a particular branch itself, pruning, or ladderizing, which basically means printing to the complete depth of the tree. So to showcase this, I'm basically going to take another case study of a gene called as APAF, which is apoptotic protease activating factor, which basically encodes a cytoplasmic protein that forms one of the central hubs in the apoptosis regulatory network. The UniProt entry for this gene is available here. You can go ahead and run a blast over it to fetch a set of sequences from which you can download how many sequences you want. You want the first 25 matches, first 50 matches, or all the matches which get generated for this particular sequence or this particular gene. Once you have that available, we are going to do the same process which we have done. That is, we have to first get the FASTA file which we can get after running a blast on this gene. Then we run a cluster-w program to get a DND file. Using the DND file, we get our Phylo.xml file by using the .convert method available in the Phylo.xml. Now, I'm going to here showcase you the processing of information from this Phylo.xml file. For that, let's first have a look at the Phylo.xml file which gets generated. It's a big file. In here, you can find out more information than what we have seen so far in any Phylo.xml. So you can see in here that this particular Phylo.xml contains information like taxonomy, ID provider, scientific name, rank. Scrolling down as we reach to more child nodes, You have got further information available in the form of name, branch length, domain architecture, scientific name, common name, detail on the sequence as well, sequence, domain architecture. So this file or XML contains or carry a lot of information regarding different clade nodes. For example, let's look at this particular clade node. So this clade node name is 9 underscore BRAFL. The branch length from the root is this. The taxonomy is scientific name, common name Florida lancelet, synonym Amphiuxus species. It also contains its sequence domain information. Okay. So now the first thing which we are going to do is we are going to understand and fetch all the terminal nodes from this file. So let's do that. So let's have a look at the program. Again, we are importing, making available the Philo and the Philo XML module available to us. Then we are again reading this XML file and creating our tree object by reading the file, this time in the XML file format, this time in the Philo XML format. Once our tree object is created, we can perform different functions on it as listed in the tree mixin module, which we just saw, right? So the first thing which we are going to do is we are going to get all the terminals. All the terminal nodes basically means all the child nodes which further do not have any more children. So we create a for loop in here for clade in tree.getTerminals. This is basically going to return to us a list of all the terminal nodes. Once we have all these clades, we are going to simply print the clade name and the clade taxonomy scientific name. Let's run this program. So we can here see that from reading this file, we have been able to make available to us all the terminal nodes information. So these are all the terminal nodes available to us followed by their scientific name. Now I can fetch more informations like this. Now this is just one file, one data set, and you can start observing the power of Biopython Biophilo module, that how easy it is to process such information and faster as well. And I can easily customize it to make it available or useful for my own needs. Now moving on, let's use a different method, is that we want to find whether this particular node or this particular gene information exists in this tree or not. Followed by we want to count how many terminal nodes were made available. So we do that by executing this program. So we get information, yes, the node did was present, and the total number of terminal nodes available for this particular phylogenetic tree is 31. Now once we are done with search and traversal, we are going to move on to the information functions. Let me comment out the functions which do not need anymore. So when we use a hash in a Python module for any of these statements, that will never get executed during the execution of this particular program. Then let's say we want to find out the distance of a node from the root. So we use the method tree.distance for this particular node. You can run a for loop in here and print the distance for all the different terminal nodes from the tree. You can find out which terminal node lies the maximum away from the tree and make assumptions or discoveries there. You can then find out the total branch length and whether the tree is bifurcating or not. So we can calculate the distance, find the tree length, and find whether the tree is bifurcating or not. Now comes some pretty interesting code, that is to find common ancestor among different clade nodes. So we are going to pick two clade nodes and we are going to find out through this phylogenetic tree whether a common ancestor is available or not. So I've picked up a node called as apaf-human and 22 underscore mouse, and I'm going to use a function called as tree dot common ancestor. So find a common ancestor for clade 1 and clade 2 nodes. So for these two nodes in the phylogenetic tree, find me the common ancestor. And we get an answer as this. This is the common ancestor of these two nodes in the phylogenetic tree. To confirm, go ahead, open the tree. You can look at these nodes and find out that indeed, yes, for these two different organisms, this indeed was the common ancestor. So, this is the name of my parent, which had APAF human and 22 underscore mouse as the child nodes. Now, let's say I want to find out another common ancestor between the parent of clade 1 and clade 2, which we have just generated, and another node. So you can do so. Let's say I want to find a common ancestor for this particular gene and the parent of these two modules. Again, I can run a tree.common ancestor function on clade 3 and clade 4. This is going to generate and find out a common ancestor between the parent of mouse and human and canfa. So the results are Eutherin is basically the class of organisms which are common parent or ancestor to these three different organisms. You can verify by reading about them. And the rest, you can go ahead, remove a particular node itself from the tree, draw the tree in different formats, prune the tree, split the tree to make only a particular branch more accessible to you. All this is very much possible with the Biophilo module. So in this particular tree diagram printing, the branch which was carrying the information regarding 12 underscore CFNA is removed. It's collapsed. Okay, so it is no longer available to us. So all this analysis by changing and making different, working with different functions of a tree mixin module is possible over a phylogenetic tree. So this was all we had for the webinar. We basically wanted to showcase to you the power of the Biopython Biophilo module, the flexibility which it offers, and the ease of use available to customize and create your own programs in Biopython to make them more reliable and secure within your own lab premises. So all this is very much possible and pretty easy to set up and do. If you guys have any questions, the forum is now open to ask questions whichever come to your mind. Please feel free to post your questions on the questions window. Okay, thanks a lot, Sebastian. Thanks a lot for joining. It was a pleasure to have you in the webinar. Thank you so much. Bye. In case you do not have questions right now, feel free to reach us at the information mentioned below, admin@scientiabio.com, and this is the contact number using which you can connect with us. Thanks a lot for your time. We hope you have liked the information shared by us. Thanks a lot for joining this webinar by Scientia Bio. Hope you have a great day ahead. Thank you so much. Bye.